Hello and welcome everyone to this podcast for cell biology. In this podcast we're going to talk about how we control cell number and cell size. I want to begin by showing these cells as they go from a fertilized egg, the zygote, to a 2-cell, to a 4-cell, to a multi-cell, to a morula, and to a blastocyst, right as it implants in the uterine wall. We've talked about these cells before, about how they don't change size and how that's related to the cell cycle. But what I want to point out here is that if we were to look at mouse cells going from zygote to blastocyst and compared that with human cells as they go from zygote to blastocyst, you wouldn't be able to tell the difference. They divide at a slightly different rate, so you would be able to tell based upon what day it was. But if you just looked at the size and their overall structure, they are indistinguishable. But we know if we were to look at an adult mouse and an adult human, we know size, shape, structure, cell number are all very different between an adult mouse and an adult human. These are all differences in cell shape, cell number. And all of this is controlled by three processes. The first is cell growth, the second is cell death, and the third is cell division. By balancing these three events, an organism controls the number of cells they have and the size of those cells. Okay, let's begin our discussion of cell death as it relates to controlling the number and shape of cells. And I'd like to begin this discussion by thinking about a community. So if you remember back to an introductory biology class, or if you took an ecology class, a community consists of all populations in a given area. So for instance, in this African savanna, you have populations of lions, populations of, I don't know if these are wildebeest, I can't tell, the picture's too small and my eyes are too bad. But you have populations of wildebeest, you have populations of antelope, you have populations of insects that we can't see, we have trees that exist in a population, the grass is here, all the other animal and plant populations here that we can't see. And they exist as this community because each of these populations rely on each other. In order for this community to persist, we have to have life and death. We have to have new organisms being born. Certain organisms are going to live longer than other organisms, but that's all needed to balance this community. And though we don't like to think about it, this lion's got to eat, and so it may eat one of these small animals over here. But that's all to balance the community. Now, I want to compare that with this human, this baby human. And animals and plants, or multicellular organisms, would be another way for me to say that, are really a community of cells. Up here was a community of different populations. Here it's a community of cell populations. You have cells that make up the liver, you have cells that make up the pancreas, you have cells that make up the eyes, you have cells that make up these very distinct tissues. Or we could think of them as cell populations in our analogy here. In order for this community of cells to survive and to persist, there has to be a balance of life and death. In order for this little baby here to survive, new cells are going to have to form, those cells are going to have to grow, but we're also going to have a decent amount of cell death. So I like this comparison between an ecological community and that of an organism, because there's these similarities between life and death balancing each other for the overall good of the community here, or the cell community in an organism. Now I'm going to focus on death for this aspect of maintaining our cell community, and I'm going to provide a couple examples of why cell death is important. We know that about half of some cells in our nervous system, or in animal nervous systems, not just ours, die during development. We also know as an adult, every 30 minutes or so, billions of cells die in our bone marrow and intestines. Some of this cell death we understand why it occurs, and some we don't understand why it occurs. But we're learning more and more about it. Because if we're just making these cells, and many of them that die seem to be perfectly healthy, but if we're just making them just to kill them, it seems energetically very wasteful. But there must be a reason. And I want to talk about a couple of examples of why creating cells just to destroy them is helpful. So examples of cell death. We know cells die sometimes to protect the organism. If too many DNA mutations are present in a cell, often that cell will be programmed to die. And that protects the whole organism because sometimes those mutations can lead to things like cancer. In development, a mouse or a human, we will have this kind of flat surface here for our limbs. So this is a very poor drawing of a limb and a future hand. And what will happen is that we will have cell death that occurs in certain regions here, ultimately to give rise to what we might recognize as a hand, or if you're a mouse, a paw, because these cells in between the digits died. Now sometimes people are born and there hasn't been appropriate cell death in between the digits. This can be surgically repaired. The next example, and probably the last one I'm going to talk about right now, for now anyhow, is the regression of a tail. Now sometimes this tail is formed because it's useful. So let me draw a tadpole. And I suspect this tadpole will be the best tadpole you've ever seen. Oh, that's awesome. But this tail here of a tadpole that's needed for its aquatic life is eventually regressed, or the cells within it die, which will lead to the fully formed frog. So I'm going to put adult frog because for everybody's safety I am not going to attempt to draw an adult frog. You'll notice that an adult frog doesn't have a tail, and the reason it doesn't have a tail is because that tail has been lost because of cell death. Now even humans during embryonic development, we have tails as well, but our tail is lost during embryonic and fetal development, and it's lost because of cell death that destroys those cells. Every now and then a human is born with a short tail, but that is usually very easily removed because usually it's just skin that's present that needs to be removed. It usually doesn't have any structures in it. Okay, I wasn't completely honest. There's one more I want to talk about, and that has to do with adult tissues remaining the same size. Now, I shouldn't say just tissues, but tissues and organs. And for this example, I want to talk about the liver, and I'm going to draw that over here. And so I'm going to draw the liver like this. Obviously, the liver is not this perfect sphere, but that's about as good as you're going to get from my drawing. But that's a liver. Now, this liver always has to remain about the same size. So here are two livers the same size. It's actually the same liver. So sometimes what could happen is liver gets smaller. It could be due to cell death of a part of the liver. It could be due to removing part of the liver for a transplantation. It could be removed part of the liver because it's diseased. But at any rate, some of that liver is removed. And this is my awesome drawing of what a liver would look like with part of it removed. Don't be jealous. It's a good drawing. And what will happen so that we can maintain this same size is new liver part is formed, all in an effort to maintain this liver. Now sometimes what can happen is the liver can get bigger. This is my example of a bigger liver. And this can happen under certain conditions, but experimentally, if we give the drug phenobarbital, the liver gets bigger. Now, when we remove the drug phenobarbital, cell death will occur, resulting in the same sized liver as we started with. So this balance between cell growth and cell death is important to maintain proper organ and tissue sizes. So I'm going to talk now more specifically. Now up to this point, I've just been using the term cell death. There's lots of kinds of cell death. One kind is called necrosis. This usually results when cells die due to an injury, some kind of trauma. When this happens, the cells will swell and burst. This results in an immune response called inflammation. As a result, neighboring cells are negatively impacted. Another kind of cell death, something that's often called programmed cell death, but we're going to use the special term called apoptosis. Apoptosis is often called a clean death. During apoptosis, a series of events occurs that prevents inflammation and damage to neighboring cells. These events include the following. The cytoskeleton is collapsed, the nucleus disassembles, the DNA fragments, because when that nucleus disassembles, various nucleases can penetrate the nucleus and begin to degrade the DNA. And lastly, the cell surface of this apoptotic cell presents receptors for phagocytes, mainly macrophages. These first three steps here, they occur within cell membranes. Now sometimes these cell membranes won't be this big cell, but rather blebs of cell that have been broken up. But they're still protected from the outside environment, the extracellular environment, because all the components are within the cells. And that is what allows for these phagocytes, the macrophages, to be recruited and engulf them. By doing this, you prevent inflammation. So let's write that down. By having these phagocytes engulf and digest these blebs of membranes containing the cell components, we prevent inflammation, and we also recycle the cellular components. So when that cell dies, it recycles those components so the rest of the cells in the organism can be used. Okay, so we've talked about cell death, we've talked about why it's important, we talked about the advantages of apoptosis or over necrosis. And now we're going to focus exclusively on apoptosis and talk about the molecules needed to cause apoptosis. And so now I'm going to add to this, it's cell death, apoptosis, and caspases. Caspases, these are the molecular proteins that regulate apoptosis. To die or to live, that's what they regulate. These caspases are proteins that would be classified as proteases. And remember, proteases are enzymes that degrade proteins. So these caspases, in one form or another, are present in our cells all the time. So how is it that our cells aren't always programmed to die? And the reason for that is that caspases are initially transcribed and translated as pro-caspases. As a general rule, whenever you see a protein with the word pro in front of it, that means it's the inactive form of the last part of that protein. So pro-caspases are inactive caspases. And let's just draw them like so. And the part here I'm drawing in orange, this is what we'll call the inactive component. It's still part of the protein. In fact, this is the N-terminus and that's the C-terminus of the protein. But this part here, encoded by the N-terminus of the protein, allows the caspase to be inactive. When the cell receives an apoptotic signal, this N-terminus part here is removed. We'll just put those up here. And the rest of the protein will bind with another activated caspase. And so now it is active and can initiate apoptosis. And what this actually does is it initiates apoptosis, but it does so by starting a cascade. And when we use the word cascade, what we mean is that this active caspase will activate other caspases. Those caspases will activate even more caspases, and so on. they are usually one directional, just like the case of this caspase cascade. Once it's activated, it doesn't go backwards. So this whole process of activating the initial caspase is highly regulated. And so what will happen then is this initial caspase, like this, and we're going to call this an initiator caspase. That's always the first one that's initiated. So they've cleverly named it initiator caspase. Once the initiator caspase is activated, it will activate several executioner caspases. These first ones might begin to degrade the nuclear lamin. Remember, this is the part that holds that nucleus together. So when this begins to degrade, the nucleus breaks down and DNA will fragment. These executioner caspases will now activate the next line of executioner caspases. These might play a role in degrading cytoplasmic proteins. These then may go on and activate even more executioner caspases involved in a different step of apoptosis. In this way, by activating this initial initiator caspase, you set the downstream events, irreversible events, to begin to kill the cell cleanly without causing inflammation. So the next question is, how are procaspases activated into caspases? All cells in a multicellular organism contain a self-destruct mechanism. Because all cells contain this, it's really important to make sure that there are many steps put in place to regulate this self-destruct mechanism. By and large, this self-destruct mechanism of apoptosis is controlled by the BCL family of proteins. These proteins either promote caspase activation and thereby promoting apoptosis, or they inhibit caspase activation, thereby preventing apoptosis. To describe how this could work, I'm going to erase this so I can have a lot of space to work on. So here's our cell. I'm going to put our nucleus over here with DNA in it, and this DNA has become mutated. Mutated in such a way that it can't be fixed. And if you remember, in previous podcasts we talked about that protein called P53. And P53 assesses whether or not that damage can be repaired. And if it is, it repairs it. If not, it signals it for apoptosis. We talked about repairing it before. Now we're going to talk about sending this cell towards apoptosis because the damage cannot be fixed. Okay, so when the DNA is damaged beyond repair, this is an apoptotic signal and it's sent to the mitochondrion. And so here's our lovely mitochondrion with the inner membrane. And if you remember, on the inner membrane there were these protein complexes called cytochrome C. Usually cytochrome C stays in the mitochondrion. That's where we want it. But once this apoptotic signal is sent to the mitochondrion, there are these proteins on the outside of the mitochondrial membrane that are called BACs or BAC. These are members of that BCL family. When this apoptotic signal signals the mitochondrion, cytochrome C leaves. I'm going to color that blue just so we can follow through here. Cytochrome C is released from the mitochondrion. Cytochrome C will then bind to an adapter protein. This is not the same adapter protein that was involved in secretion. This is a different adapter protein. So now this adapter protein is activated and it will begin to assemble the apoptosome. And the apoptosome will contain 7 of these activated adapter proteins. And they can only form when cytochrome C is bound to the individual adapter proteins. Next, what happens is procaspase 9 comes in. This is an initiator caspase. and it will bind to these adaptive proteins in this apoptosome. And it will bind in between each of these adapters. So there will be 7 of them as well. And now it is an activated apoptosome, as well as it contains activated caspase 9s. This now will go on to initiate caspase cascade by activating the executioner caspases. So I don't think I said it before, this is an example of an intrinsic apoptosis, because this occurs within the cell because of signals received within the cell. So it's intrinsic. I want to spend just a little bit of time talking about an extrinsic apoptotic pathway. So extrinsic. This occurs when you have a cell here and it receives an apoptotic signal from the outside of the cell. In this case, it's going to be a killer lymphocyte, which is part of our immune system. It has detected something on this cell that has told the organism that this cell must go through apoptosis. And I'm not going to go into great detail here, except to say that this killer lymphocyte will bind to this cell, and not anywhere, but specifically to these receptor proteins called the FAS death receptor. And if you're a cell, you never want this to be bound, because when this gets bound, this will set into motion the activation of initiator caspases. This in turn will activate executioner caspases and ultimately lead to apoptosis. So this is extrinsic apoptosis. Now we just got done talking about the signals that a cell can receive intrinsically or extrinsically to cause cell death. But I want to now talk about extracellular signals for cell growth and survival. So for the cell to grow and survive, it can receive one of 3 signals. It can receive a survival signal, and essentially what this does is it represses apoptosis. These extracellular signals can be mitogens, and we've already talked about mitogens and how they signal for cell division. We're not going to talk any more about those in this podcast, but we will talk about signaling, survival signals, and this third one here, growth factors. And growth factors stimulate cell growth. And cell growth means both in size and mass. But let's first start talking about survival signals. So here's our cell here, and if this cell is signaled to survive, it's going to bind to its outer surface here a survival factor. An example of a survivor factor is GDNF. GDNF stands for glial-derived neurotrophic factor, but you just need to know GDF. So it binds here, and when it binds, it will activate an intracellular signaling pathway and activate a specific protein that we're not going to worry about the name of right now. But that will in turn move into the nucleus. Here's our nucleus. And it will activate the transcription of various genes, but the most important one right now is the gene called BCL2. With a name like that, it's got to be in the BCL family, and of course it is. And this BCL2 gene, when activated, will make the protein BCL2. And BCL2 functions at the mitochondrion. And at the mitochondrion, mito for short, it inhibits Bax protein. Remember, Bax is the one that will allow cytochrome C to escape. So BCL2 blocks it, so it can't do its job. And by doing this, the cell will not go through apoptosis. The only way it can go through apoptosis at this point is by inactivating BCL2. And when that happens, cell death can occur. So any cell that needs to survive, and maybe not divide, maybe not grow, just survive, like say our neurons, will receive these these survival factors, which will then express BCL2. Next, let's talk about growth factors. Remember when we talked about our cell cycle, that at the end of this cell cycle, a new cell was produced. This cell, just left to its own devices over time, would get smaller and smaller and smaller. But it maintains this size due to a balance of protein synthesis and protein degradation. But if a cell is to get bigger, what has to happen is protein synthesis must be increased and protein degradation must be decreased. Usually it's a balance, but to get bigger, protein synthesis goes up and protein degradation goes down. Now, to accomplish this, let's draw our cell over here and make it extra big. It shouldn't be too surprising that on this surface here, there's a receptor that will bind a growth factor that I'm just going to abbreviate GF. Upon binding that growth factor, various intracellular pathways are signaled. Protein synthesis increases and protein degradation decreases. The end result is a larger cell. Okay, that's all I have to say for this podcast on cell growth, cell death, and cell survival. If you have any questions at all, please make sure you come see me. If not, I'll see you in class. Bye.
You're still here? It's over. Go home.